Hello everyone, my name is Fabiana. I'm a PhD student with Dr. Phil Cardoso at the University of Illinois, and today I will be talking about the effects of non-toxigenic Clostridium oral supplementation on production, health, and metabolism of Holstein cows. So just to give a brief summary about our experiment, we supplemented the cows for 70 days with non-toxigenic Clostridium. And the non-toxigenic Clostridium supplementation did not alter physiological parameters in dairy cows. However, the non-toxigenic Clostridium reduced the gene expression of TLR2, that is an inflammatory gene in the rumen papillae. So as outlined, I will give an introduction, and then objectives, material and methods, results, and then finally the conclusions. So to better understand the idea besides this experiment, I would like to start to talk about the microbiome. What is the microbiome? It's the microbial makeup of all the existing microorganisms like bacteria, yeast, mold that you can find in the farm environment. Once you understand the microbiome at the farm, you'll be able to control it and improve the health of the animals. The focus here is to produce safe food in an environment that does not contaminate the animals and consequently does not affect the product quality. So due to the microbiome, we can tell that the Clostridium contamination is a reality in feeds used for animal diets and in the farm environment. The occurrence of the Clostridium in the dairy farm is a big concern for the producers because the spores can contaminate the soil, animal feed, TMR, and feces, which could contaminate raw milk via dirt contamination during milking and from cow's diet. However, the microbiome will be different between farms in different states and even farms that are next to each other, which makes necessary the evaluation of each farm individually. This means that the same system in one farm may not work the same in another farm, due to the difference in the farm's microbial environment and challenges. So to better understand it, March et al. 2018 quantified total Clostridium load in samples as TMR, corn silage, and haylage across the United States. And they reported that the most prevalent Clostridium they found was Clostridium perfringens, and 88% of the Clostridium perfringens isolated was identified as toxin type A. In addition, the two most prevalent Clostridium found after perfringens was bifragmentans, representing around 13% of the total Clostridium found, and Baerenkii, representing 10%. These both, they are classified as a non-toxigenic Clostridium, so doesn't cause the disease that the Clostridium perfringens cause in dairy cattle. So the objective of the experiment was to evaluate the effects of non-toxigenic Clostridium oral supplementation and its metabolic end products of fermentation on production, health, and metabolism of Holstein cows. So as a material and methods, the experimental period was 70 days. We used 41 multiparous Holstein cows. The cows were in the tie stall barn, in average had 192 days in milk, third lactation, producing around 23 kilograms of milk a day. In the experiment, we had 11 blocks and 4 treatments. So the treatment were control with no Clostridium supplementation, low, medium, or high Clostridium supplementation. So the treatment were given to the cows twice a day. The first time in the morning as bolus. The treatment was placed in a gelatin capsule and given to the cows using a gun that was introduced in their mouth, as you can see in the first picture. And the second dose was in the afternoon. The treatment was mixed with ground corn and given to the cows as a top dress right after we feed the cows, as you can see in the second picture. So here you can see the figure here shows that the Clostridium count in the TMR sample per week. Once a week, the TMR samples were sent for Clostridium analysis. So the Clostridium dose that we gave to the cows was 1 log higher than the average levels detected in the TMR content samples, and cows were divided into 4 treatments to contain 0 Clostridium supplementation, that is the control, low, medium, or high Clostridium supplementation. So the samples were taken daily, for example, milk twice a day, dry matter intake, treatment, sign of diseases. Also weekly, we collected data for body weight, body condition score, fecal grabs, and pH. And every 2 weeks, we collected milk samples, blood samples, rumen fluid, and rumen pH. So in the last day of the experiment, 15 cows were used for rumen papillae collection for analysis of gene expression. So the statistical analysis, the data collected from week 1 to 9 were analyzed using the procedure mix of SAS, and then the adaptation phase was used as a covariate for all the variables. In the adaptation phase, the cows did not receive any treatment. So the mixed procedure of SAS was used to model the fixed effects of treatment and block. Cow was the experimental unit and considered as a random effect. So as a result, here in the first table, you can see the ingredient composition of the lactation diet fed to all the cows during the experimental period. In this experiment, all the cows received the same diet with 40% of corn silage as dry matter. The second table here, you can see the mean chemical composition and associated standard deviation for the diets. And here, these diets were formulated to be according to the diets that people feed in the Midwest, like with 16% of crude protein, 30% of NDF, 27% of starch. So here's the dry matter intake. The high Clostridium supplementation tend to increase dry matter intake. We postulated that the non-toxigenic Clostridium could have an impact in the toxin produced by perfringens, causing an increase in dry matter intake. And however, we didn't have any difference in milk production, as you can see in the figure number 2. So this graph shows that we did not find any difference between treatments, but the percent of fat and proteins in normal range. Cows in control tended to have increased urea and nitrogen in milk compared to cows in medium. However, urea and nitrogen in milk values for all the treatments are in the normal range, that is in between 10 to 14 milligrams per deciliter. In these figures here, we did not, in the first one, you can see the rumen pH. We did not see any difference between treatments for rumen pH. However, we report a treatment week interaction where you can see that control cows most of the time had a higher rumen pH compared to others. In the second picture, we reported a tendency for fecal pH where control had a higher fecal pH than low. And also we found a treatment week interaction where you can see that during week 1 to 3, control cows had a higher fecal pH compared to others. So here is the gene expression results. Cows in control had increased TLR2. Previous studies have suggested a relationship between the upregulation of this gene in rumen papillae with an increase in immune system. So in the current experiment, the expression of the gene TLR2 was lower for cows. As you can see here, was lower for cows supplemented with Clostridium compared to control cows, which could be an indication of a less healthy epithelial barrier. So in the first figure, cows in high tended to have an increasing CLDN1 compared with cows in medium. In the second picture, figure, cows in low and high had a greater increase in CLDN4 compared with median. So these genes in previous research showed that in lactating Holstein cows, both CLDN1 and 4 expression were upregulated in rumen epithelium with the lower pH of induced acidosis challenge. However, it's unclear how the Clostridium supplementation would influence anti-junction response. So finally, cows in control and median had greater increase in HMG-CS2 compared to cows in low and high. These genes plays an important role in the metabolism of VFA in the rumen epithelium and is associated with changes in VFA levels. For example, when rich forage diet is changed to rich starch diet. However, in the current experiment, we did not report any difference in VFA concentration. So now the conclusions. Cows were supplemented with non-toxigenic Clostridium for 70 days and did not alter the physiological parameters in dairy cows. Cows in high Clostridium supplementation tended to have an increase in dry matter intake. Cows in control tended to increase the urea and nitrogen in milk. However, and then the non-toxigenic Clostridium reduced the expression of the TLR2 inflammatory gene in rumen papillae. So I want to thank you all for watching, and I will be happy to answer any questions. Thank you.